Okay, so we went from the right atrium down to the right ventricle, right through the tricuspid valve. The right ventricle contracts, and then we're going to go from the right ventricle where? In general. Where does the right ventricle pump to? The lungs. Right, so the right ventricle contracts, and that sends blood into the pulmonary trunk, right? And then ultimately to the lungs, right? We want to go through the pulmonary circuit. The pulmonary trunk, remember, is that really big artery that leads out of the right ventricle, and it kind of curves up over the heart, and then remember it splits, and it goes to the left and the right, because we've got to go to both lungs. So it divides into the left pulmonary arteries and the right pulmonary arteries. And again, that brings the blood to the lungs. Why? To get oxygen, right? To give away CO2 and to pick up oxygen. As the blood flows from the right ventricle into the pulmonary trunk, it flows through another one of our valves, right? The pulmonary semilunar valve. So if I were looking atrium, ventricles, I'm going out through the pulmonary trunk. So the pulmonary semilunar valve would be right there, right? Between the right ventricle and the pulmonary trunk. When the ventricle contracts, this valve is open so that blood can be ejected from the ventricle. When the ventricle relaxes, remember the blood flow coming back shuts that valve, and that prevents the backflow of blood into the ventricle when it's relaxed. Okay, so the blood cannot flow backwards from the pulmonary trunk into the ventricle. So it's open when the ventricle contracts, closed when the ventricle relaxes. So you guys will notice it's kind of opposite with the AV valves and the semilunar valves. Right? When the semilunar valves are open, the AV valves are closed. When the AV valves are open, the semilunar valves are closed. We'll kind of look real quick. So this is showing us when the ventricles are contracting. We're looking down the ventricles. So it's like if the top of the heart was here and we sectioned it. Okay, so we kind of took the atria off and we're looking down at the top of the ventricles. So you see the ventricles, right? These are showing you the AV valves. So the atria would be right on top right here. So when the ventricles contract, that closes the AV valves because we don't want the blood to go backwards from the ventricle up into the atria, and that pressure opens the semilunar valves so that we can eject blood, right? Because remember, that's the whole point. The ventricles contract, we want to pump blood out. So we open up the aortic valve and the pulmonary valve so that blood can leave the ventricles and go out through the arteries to wherever it needs to go. Alright, so we just went to the lungs to get oxygen, and as the blood travels back towards the heart, it collects into the two left pulmonary veins and the two right pulmonary veins. Remember, we have a superior and an inferior pulmonary vein on each side. Pulmonary because they're coming from the lungs, veins because they're heading back towards the heart. So pulmonary veins are going to dump what kind of blood? Oxygenated or deoxygenated? Oxygenated blood. Right, we just got the oxygen from the lungs. So we're going to dump that oxygen-rich or oxygenated blood into the left atrium. Right, that's a receiving chamber, receives from the pulmonary circuit. From the left atrium, blood's going to drop down where? From the atrium, left atrium, into the left ventricle. And as it does that, it passes through another AV valve. What's the AV valve called on the left side? Bicuspid, or clinically we say the mitral valve. So blood is dropping down from an atrium to a ventricle, right? From the left atrium down to the left ventricle. It's going through the bicuspid valve or the mitral valve. This little opening right here. So this valve must be open when the ventricle is relaxed, right? So that the ventricle can fill with blood. And then again, just like the right side, remember when this ventricle contracts, this valve gets pushed up because we don't want what to happen? Backflow. We don't want the blood to go from the ventricle back up into the atrium. Now, when we look at this bicuspid valve, we see that it is quite a bit stronger than the tricuspid valve. Why do you think that is, that the valve on the left side would be stronger than the valve on the right? What's up? The left side's bigger. Why? Got to push blood through the whole body. Remember, the right side, the right ventricle just has to pump to the lungs and back. And pretty strong. The left side has to pump to the entire body, including all the way down to your big toe and all the way back again. That takes more force. So the left side is a stronger ventricle that contracts harder. So the left AV valve, the mitral valve, needs to be a stronger valve. And that's why there's that structural difference between the two sides as well. This is kind of showing us that. So this is showing us a schematic of when the ventricles are dilated, when they're relaxed and open, the heart's not contracting. You can see the left ventricle has a thicker, stronger wall than the right ventricle. And this here is just showing you when the heart's contracting. Again, you can see that this has a lot more thickness to it, right? And as it's going from this volume to this volume, that's a lot of force that it's going to contract with. To push the blood out through the whole systemic circuit. So the left side is bigger, more massive, and contracts harder. Coronary circulation, guys, again, are just the vessels that supply blood to the actual myocardium, right, to the actual heart muscle itself. So we take oxygenated blood and deliver it to the heart muscle via the coronary arteries. And then we collect that deoxygenated blood from the heart and end up dumping it back into the right atrium via the cardiac veins. These next few slides we'll actually talk about in lab tonight because these are just their lab things. Like going through these pictures and identifying them we'll do in lab. They're not things that we actually need to do much. Okay, I'm gonna get through like 10 minutes of part 2.
More slides on part 1, right?
For the coronary?
Yeah.
So coronary arteries, left coronary, right coronary, then the cardiac veins, and then a couple pictures.
Okay, so
Yeah, yep, those are all lab things. Okay guys, so we're going to use the last 10 minutes or so to just introduce the heart's conduction system or conducting system. Before we do that, I just kind of want to review a tiny bit about resting potential, transmembrane potential, what the resting potential of a cardiac cell is, because that's going to be super important. You will not understand action potentials, you won't understand anything about the conduction system if you don't understand this stuff. So it kind of relates back to when we talked about muscles and neuro and all the ion movements. So when we look at a cardiac muscle cell, hey, here's my cell. Right off the bat, remember we have differences in ion concentration inside a cell and outside a cell. What ion do we have more of inside? If you had me, you better speak up. Thank you. What ions do we have more of outside? Sodium. Sodium? Calcium? Who's that little negative guy? Good job. And chloride. Overall, is inside the cell more negatively charged or positively charged? Negatively charged, right? Because of all those negatively charged proteins that are stuck in there. So inside, when we look at a cardiac muscle cell, the resting potential, so the charge at rest, is about 60. Well, when we're looking at our conduction system, the charge is about 60, negative 60 millivolts, okay, for our conduction system cells. The threshold voltage, okay, the voltage we have to get to generate an action potential, right, this electric charge that we can then spread, is negative 45 millivolts. So how do we get there? What do we do to change from negative 60 to negative 45? Normally, just in general. We allow the cations to enter, right? We need some positive charges to enter into the cell, right? And then it's less and less and less and less negative. That depolarizes the cell. We want to allow that to happen until we get to threshold. Remember, the key with threshold is that opens our voltage gated channels, right? Once you get to that threshold voltage, all those voltage gated channels open, and it's like rapid depolarization, right? That's your action potential. So just kind of remember those kind of key things from A&P1. If any of that is not super easy and familiar, we need to talk at some point. I do have neuro lectures, like videos posted, that you can look at those too, if you want to review it, but we need to chat at some point soon if that's not all super familiar, okay? Okay, so that's just kind of background. We're going to talk about the conducting system or the conduction system. A heartbeat, that right, that you can hear and feel, is one full cardiac cycle. Okay, so a cardiac cycle is that the two sounds. What happens during that heartbeat is all of the chambers will go through this series of contraction and relaxation. Contraction, relaxation. So the atria do this first, the two atria at the same time contract and relax. Then the two ventricles do it at the same time. They contract and then they relax. That's a heartbeat. That's a cardiac cycle. When we look at this occurring, when we look at this heart contraction occurring, we see that there are two different types of cardiac muscle cells that are involved. Okay? We have the contractile cells. Those are the normal muscle cells that contract, right? They're the ones that do the work, the ones that make up that thick layer of cardiac muscle, like in the ventricles that squeeze and contract and pump blood. But we also have a highly specialized type of cardiac muscle cell that makes up the conducting system, or the conduction system. You can think of this as the electrical system of the heart. Okay, the conducting system is the electrical system that initiates and then spreads or distributes the action potential, which remember says contract, right? That's what makes the muscle actually contract, is the action potential spreading. So the conducting system actually starts that action potential and then it spreads it throughout the heart. So it controls the heartbeat. It tells the heart when to contract, in what order to contract, how fast to contract. Okay, so the conduction system actually controls the cells that are doing the contracting. Did you guys have another slide in there? I did. That's weird. Okay, so when we look at the heart is super cool. The heart displays something called automaticity or autorhythmicity. Right? Auto means self. And think of like rhythmic as something that's right like a rhythm. So the heart has its own rhythm that it generates. The heart has the ability to automatically generate action potentials to contract. It does not need a neuron. It does not need a hormone. It does this itself. That's very different from skeletal muscle. Remember skeletal muscle, you had the motor neuron, right? Your synaptic cleft, and then your muscle fiber. And if this motor neuron did not release the acetylcholine to stimulate the muscle fiber, that muscle did not contract, right? You needed it to be stimulated by a neuron. Cardiac muscle does not need a neuron to tell it when to contract. Okay? Neurons can alter it, but it tells itself. It sets its own base rhythm. It has automaticity. So the reason for this is because of its highly specialized conduction system or conducting system. Again, I told you guys that this was like the electrical system of the heart. This is the system that starts that action potential and spreads it throughout the heart in a very specific pattern. We'll see that there's a bunch of different kind of pieces involved with this. The sinoatrial node, which is one of the most important parts, is at the very top of the right atrium. We'll have some pathways that interconnect these two nodes. And then we have the atrioventricular node. It's kind of in like the floor of the right atrium, right by where we enter into the septum. Then we'll see that we have a bunch of different pathways. All of these conducting cells or electrical cells are kind of grouped together and organized into different pathways that spread throughout the myocardium. I'm gonna take like 2 more minutes, okay? Your teacher at 7 will forgive me. If you have another class, you can go to it. I won't get offended. So, the SA node, we said is up here, the top of the right atrium. The AV node is down here by the floor of the right atrium, right by the septum. Then we have conducting cells arranged in all of these pathways. Okay, so like all the other electrical cells are arranged in all these nice little pathways that go throughout the heart. Up here in the atrium, we have internodal pathways, internodal, between the nodes. So from the AV node from the SA node to the AV node. So these are the internodal pathways. We've got all these electrical cells arranged in between these two nodes. Then when we look down to the ventricle, we have the AV bundle. Okay, so from the AV node, the AV bundle just enters right into the septum. Then the AV bundle splits into the bundle branches. They just go down either side of the septum. Again, these are just electrical things, right? So we're just generating these electrical currents, the stimulus, and we're spreading it throughout the heart. At the very bottom of the heart, we'll see that we have Purkinje fibers. So all these little guys down here, those are showing us Purkinje fibers. What's going to happen here, just in general, we're going to start the impulse, and we're going to spread it to the atrium as we head down here to the AV node. We're going to pause. Then we're going to enter the septum. We are not going to stimulate the ventricles yet. We're going to travel down the septum to the apex. We're going to curve around. Then we're going to stimulate the ventricles from the bottom up towards the top like this. Okay, that's kind of the general gist of what we're going to do. Let's do this slide and then I'll stop because this is going to introduce kind of an important concept to you that I want you to marinate and kind of spend some time with before this class. You gotta love my analogies. Okay, so the SA node and the AV node. Remember, if this is my heart, the SA node was right here, the AV node was right there, right? That would be right there. So we said the impulse goes like that, and then it comes down and it goes around. The cells that make up the SA node and the AV node are highly specialized cells. They have something called a pre-potential or a pacemaker potential. When we look at these cells, they do not have a stable resting membrane potential. Most cells do, right? Most cells sit at negative 70 millivolts. That's their resting potential. Or negative 90 millivolts. That's their resting potential. The cells at the SA node and AV node do not have a stable resting membrane potential. So without anything happening, without any stimulus coming in, without opening any channels, nothing happens, and they gradually depolarize towards their threshold voltage. At rest, normally, they just creep up towards threshold voltage. So looking at the cell, so say, here's my cell, and I'm sitting at negative 60 millivolts. And I have to leak all the way up to negative 45 millivolts before I get to my threshold voltage and I open up all of my voltage gated channels and it's like done, right? Action potential created. So when we look at these cells that are present in the SA node and the AV node, we see that their sodium channels are a little funny. Their sodium channels are extra leaky. So their sodium channels allow sodium to flow more than other cells do. And where is there more sodium? Outside. So diffusion says the sodium's going to go where? Inside. So as positive charges leak in, we get less and less negative, right? If there's a difference between inside and outside of 60 millivolts, and now I allow, to make it easy, we'll say 10 positive charges in. Now I'm not at negative 60, I'm at negative 50, right? And then I allow another, that's not really the way it works, but, and then I allow another 5. Now I'm not at negative 60, I'm at negative 45. Right? So as these positive charges leak in, they make this less and less and less polar. They gradually depolarize the cell to threshold because sodium allows sodium to enter the cell. This happens all the way until we get to depolarization, and that opens the voltage gated channels and it generates an action potential. This is why they don't need a neuron to turn them on. They turn themselves on. Okay? So, the rate of depolarization differs between the SA node and the AV node. They both have the ability to do this. They can both leak to threshold and generate their own action potentials, but they do it at different speeds. The SA node up here is leakier, if you will. The channels allow sodium to enter at a faster rate, so the SA node depolarizes at a faster rate. The SA node typically depolarizes between like 80 and 100 times per minute. It leaks quicker, gets to depolarization faster. Because of this, the SA node is what we call the pacemaker of the heart. It sets the pace that your heart is going to beat. Okay? Because when it fires, remember those gap junctions mean that that's going to spread to everyone, right? So once this guy fires, everything fires. So if this guy's firing at 80 times a minute, the whole heart's beating at 80 times a minute. If this guy fires at 100 times a minute, the whole heart's going at 100 times a minute. Everything follows suit. Now, the AV node, again, can leak to depolarization. It can spontaneously depolarize on its own. It does it slower. Sodium does not enter as quick. So it depolarizes between 40 and 60 times a minute. Now, in reality, it doesn't actually ever have the chance to do this because the SA node stimulates it before it ever leaks on its own. But if for some reason we cut it off and the SA node were no longer stimulating it at 80 times a minute, this could actually generate action potentials and we could start the heart rate right here instead of right here, which happens in some conditions as we'll see later. Right? It's just the heart rate would be much lower because now instead of relying on this guy that's really leaky and beats nice and quick, we're relying on this one that say maybe can only depolarize 50 times a minute. So now the heart follows suit and only beats 50 times a minute. Is that okay? Okay, so steady action potentials, steady ion movement, steady channels. Email me or stop at my office if you have any questions. Thank you for bearing with me today. We'll see you guys in lab. If you're not there by 7, you're in trouble.